The Triticeae tribe within the Pooideae subfamily of grasses includes genera with many domesticated species. Major crop genera found in this tribe include wheat, barley, and rye. Crops in other genera include some for human consumption and others used for animal feed or rangeland protection. Among the world's cultivated species, this tribe has some of the most complex genetic histories. An example is bread wheat, which contains the genomes of 3 species, only one of them originally a wheat Triticum species. Seed storage proteins in the Triticeae are implicated in various food allergies and intolerances. Genera of Triticeae. This list of tribes broadly follows that in grass genera of world. Although taxonomic disagreements exist about the precise circumscription of some genera, this scheme is typical of those used in taxonomic literature. Aegilops, Agropyron, crested wheatgrasses, Amblyopyrum, Anthocyaton, Ostropyrum, Cocchanea, Crythopsis, Dasypyrum, Elymus, Texas ryegrass, etc., Elytriga, Eremium, Eremopyrum, Festucopsis, Hainaldia, Inradia, Heteranthelium, Hordelimmus, Hordum, Hystrix, Kengilia, Lamas, Lophopirum, Malacarus, Pascopyrum, Western wheatgrass, Peridiction, Psathyrostochis, Pseudoroephneria, Sicale, Satanian, Stenostochis, Teniatherum, Thanopyrum, Triticum. Cultivated or edible species. Aegilops various species occur in pre-agrarian archaeobotanical remains from Near Eastern sites. Their edible grains were doubtless harvested as wild food resources, speltoids, ancient food grain, putative source of B genome in bread wheat and G genome in T. Timofeeva, Torski, source of D genome in wheat. Amblyopyrum Muticum, source of T genome. Elymus various species are cultivated for pastoral purposes or to protect fallow land from opportunistic or invasive species. Canadensis, edible, bread flour capable, fiddly seeds, Trachycaulus, pastoral cultivar. Hordum many barley cultivars, vulgar, common barley, bulbosum, edible seeds, murinum, cooked as pear plus or minus ole, bread flour capable, medicinal, diuretic. Lamus arenarius, bread flour capable, possible food additive, race mosses, drought-tolerant cereal, used in Russia, condensatus, edible seeds, harvesting problematic small seeds, triticum, used in North America, seed hairs must be singed. Sicale rise, cereal, livestock feed and sourdough bread, 6 subspecies. Cornuta herbal medicine at very low doses, deadly poisonous as food. Strictum, actively cultivated, sylvester actively cultivated in Tibet and China highlands. Vavilovia, edible seeds, thickener. Triticum wheat, Aestivum, compactum, maca, spelta, sphaerococcum. Monococcum, Timofeeva, Turgidum catholicum, dicocoids, dicoccum, used to make farro, durum, paleocolchicum, polonicum, tyrannicum. Genetics, Triticeae and its sister tribe Bremoan join former sister clade with Pooideae and Avenea. Intergeneric gene flow characterized these taxa from the early stages. For example, Pooideae and Avenea share a genetic marker with barley and 10 other members of Triticeae, whereas all 19 genera of Triticeae bear a wheat marker along with Brema. Genera within Triticeae contain diploid, allotetraploid and or allohexaploid genomes. The capacity to form allopolyploid genomes varies within the tribe. In this tribe, the majority of diploid species tested are closely related to Aegilops. The more distal members include Hordum, Eremion, Psathyrostochis. The broad distribution of cultivars within the tribe and the properties of the proteins have implication in the treatment of certain digestive diseases and autoimmune disorders. Evolution of the tribe, one of the earliest branches in Triticeae, the Pseudoroegeneria, produces the genome STST and another Hordum then genome equals HH. Allotetraploid combinations of Pseudoroegeneria and Hordum are seen in Ulmus, but also shows introgression from Australian and Agropyron wheat grasses. Elymus contains mostly Pseudoerogeneria mtDNA. Many genera and species of Triticeae are allopolyploids, having more chromosomes than seen in typical diploids. Typical allopolyploids are tetraploid or hexaploid, XXYY or XXYYZZ. The creation of polyploid species results from natural random events tolerated by polyploid-capable plants. Natural allopolyploid plants may have selective advantages and some may permit the recombination of distantly related genetic material. Pooideae wheat is an example of a stable allotetraploid wheat. The Triticeae may be a very early branch from the goat grass clade, as branch these are almost contemporary with the branching between monoploid wheat and Aegilops torschi. Studies in Anatolia now suggest rye was cultivated, but not domesticated, prior to the Holocene and evidence for the cultivation of wheat. As climate changed, the favorability of Triticeae declined. At that time other strains of barley and wheat may have been cultivated, but humans did little to change them. Goat grasses and the evolution of bread wheat. Tetraploidation in wild emmer wheat Aegilops appears to be basal to several taxa such as Triticum, Ambulopirum, and Crythopsis. Certain species such as Aegilops beltoides could potentially represent core variants of the taxa. The generic placement may be more a matter of nomenclature. Aegilops and Triticum genera are very closely related, as the image to the right illustrates. The Aegilops species occupy most of the basal branch points in bread wheat evolution, indicating that Triticum genus evolved from Aegilops after an estimated 4 million years ago. The divergence of the genomes is followed by allotetraploidation of a speltoid goatgrass basal wheat species Triticum boeticum with strains in the Middle Eastern region giving rise to cultivated emmer wheat. Hexaploidation of tetraploid wheat hybridization of tetraploid wheat with E. Torschi produced a hulled wheat similar to spelt, suggesting T. spelta is basal. The torschi species can be subdivided into subspecies Torschi and strangulata. The D genome of bread wheat is closer to AT strangulata than AT torschi. It is suggested that T. torschi underwent rapid selective evolution prior to combining with tetraploid wheat. Wild Triticeae use by humans. Intense use of wild Triticeae can be seen in the Levant as early as 23,000 years ago. This site, Uala 2, also shows that Triticeae grains were processed and cooked. Many cultivars appear to have been domesticated in the region of the Upper Fertile Crescent, Levant and Central Anatolia. More recent evidence suggests that cultivation of wheat from emmer's wheat required a longer period, with wild seeding maintaining a presence in archaeological finds. Pastoral grasses. Triticeae has a pastoral component that some contend goes back to the Neolithic period and is referred to as the garden hunting hypothesis. In this hypothesis, grains could be planted or shared for the purpose of attracting game animals so that they could be hunted close to settlements. Today, rye and other Triticeae cultivars are used to graze animals, particularly cattle. Rye grasses in the New World have been used selectively as fodder, but also to protect grasslands without the introduction of invasive Old World species. Triticeae and health. Glutens in the Triticeae tribe have been linked to gluten-sensitive diseases. While it was once believed that oats carried similar potentials, recent studies indicate that most oat sensitivity is the result of contamination. Triticeae glutens examines of the proteins of Triticeae, important in the link between gluten, gastrointestinal, allergic and autoimmune diseases. Some of the recently discovered biochemical and immunochemical properties of these proteins suggest they evolved for protection against dedicated or continuous consumption by mammalian seed-eaters. One recent publication even raises doubts about wheat's safety for anyone to eat. Overlapping properties with regard to food preparation have made these proteins much more useful as cereal cultivars, and a balanced perspective suggests a variable tolerance to Triticeae glutens reflects early childhood environment and genetic predisposition. References. External links: PubMed, Triticeae, Database of Edible Seed Plants, International Center for Agricultural Research in the Dry Areas, an excellent resource for the ancestral genetics of Triticeae. Aegilops Comparative Classification Table, Triticum Comparative Classification Table, Genomes in Aegilops, Triticum, and Amblyopyrum, Triticeae germplasm.